Last week we learned about how to insert a gene into a plasmid and then feeding that plasmid into bacteria so that they could mass produce whatever the protein is encoded by the gene, right? And in the process, we learn about a few techniques. We learn about PCR, which is used to amplify genes, right? We can use PCR to copy a gene many, many times. We learned about restriction enzymes, which allows you to cut genetic material, cut DNA at very specific locations. And then we talked about transformation, which is feeding the plasmid to the bacteria. And more importantly, how to distinguish bacteria that has accepted the plasmid versus those that rejected the plasmid. Okay. So we are almost done with the biotech stuff. There are a few more techniques we are going to learn today, starting with DNA fingerprinting. Okay. So you might have heard of DNA fingerprinting before. It's used to for various applications. Okay, it can be used to determine relationship between individuals. Right? Like if they're adults, like who the parents are, you can get a sample from the child and then get a sample from either the father or the mother, and then you can check for relationship of long lost family members, or if they're concerns about who's the father, you can do DNA fingerprinting. DNA fingerprinting is also commonly used for crime scene investigation. If they find like a sample of a suspect at the crime scene, and then you have a bunch of people and you want to know if they're there as well, you can do DNA fingerprinting. It's more accurate than traditional fingerprinting, okay? Because now we are analyzing the DNA profile of the individual. So let's take a closer look at how that works. Now, there is a big word. It's polymorphism. Polymorphism, it's basically any difference you can detect between the DNA of individuals. Okay, so if you look at your DNA, you look at my DNA, any detectable difference between the two samples is a polymorphism. Remember, even though we have different ethnicity, and we are very different, we're not related, but our DNA are 99.9% identical, right? So the 0.1% difference that distinguish you from any other person on the planet, that's basically the polymorphism that we are talking about. Okay. So what kind of differences are we detecting? Well, one type of polymorphism that we're particularly interested in when it comes to DNA fingerprinting is something called short tandem repeats, or STR. STR. Tandem means one after another. And so they are repeats that goes one after the other, and they are often very short, usually 2 to 3 nucleotides. For example, we have a CAG, 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 CAG. So this is a triplet that's repeated multiple times. And so because this one is repeated 4 times, we would write it as CAG 4. Okay? So where is the polymorphism? The difference, right? The difference is not the kind of repeats that you have or the kind of repeats I have. Okay? We all have the same repeats at the same location in our genome. So if I have CAG repeated 4 times on my chromosome 16, you will also have CAG repeated on your chromosome 16, but the difference is the length of the repeat. Okay, so I might have it repeated 4 times, you might have it repeated 10 times. Okay, people who are related often have the same number of repeats or similar number of repeats. Okay, so these are STR, or short tandem repeats. Some of these STRs are related to certain diseases. For example, Huntington disease is a neurodegenerative disease similar to those of like Parkinson, Alzheimer's, right? And there is a gene that is strongly linked to the Huntington disease. It's the Huntington gene, HTT. In this gene, there is STR in it, CAG, the one I told you about. Now, if it's repeated 10 to 35 times, that's normal in the general public. However, anything longer than 35 is going to give the person Huntington disease. Right? The thing about short tandem repeats is when the DNA polymerase tries to copy your DNA and it gets to one of these repeats, because it's repeated so many times, it's very easy for it to make a mistake. Right? Sometimes it would add like an additional copy to it, or sometimes it would miss a copy. So let's say there's 10 CAG repeated. When you copy the DNA, it's not uncommon for the polymerase to additionally add extra one. So it becomes CAG 11, okay? Or miss one, so it becomes CAG 9, right? So what happens is once this CAG on the HTT gene becomes too long, then the person has Huntington disease. Huntington disease is a dominant condition. So, if the parents has it, there's a 50% chance that it's going to be passed on to the next generation, assuming that they are heterozygous. And what happens is the disease actually don't manifest itself. It doesn't actually show up until later on in life. And by then, people usually already have kids. So, the kids find out that one of the parents have Huntington, then they have a difficult choice, right? Do they do genetic testing to find out whether they have the gene or not, right? So some people might want to know because, if they actually have the gene, that means they will definitely have the disease later on in life, and they might not want to have kids, right? Because, it's 50% to pass on. But, some people don't want to know, right? Because then, you always have this thing hanging over you, right? It's knowing that you're going to have a disease that has no cure is not an easy thing to cope with. Okay, so there's a little bit of a dilemma there. Okay, that's Huntington disease. It basically you will lose control of your muscles and become paralyzed, and there's no cure for it. People will die from this. All right, but not all STRs are necessarily associated with diseases. Okay. That was just like an example I wanted to show you. Much of the STR that exists in our body are not associated with diseases. And there are many STRs scattered throughout our genome. All right. So for example, person number 1 might have AGAT repeated 12 times. For another person, it's only repeated 5 times. So how long is this STR? 4 nucleotides repeated 12 times. It's how long? 48, that's right. 4 times 12 is 48, right? But for this person, it's 4 nucleotides repeated 5 times, so that's only 20, right? So this is longer than that. Do you guys understand? Which one is long? Oh, that's a dumb question. They are the same. Which one is longer here? Top or bottom? Top. So you see, if I'm able to find a bunch of STRs, that means the length would be like long, short, short, long, short, whatever, right? Like each person would have a very distinct combinations of these DNA lengths. And if we can find a way to separate the DNA based on the size, then we can create a unique barcode for each person based on their STR length. Okay, I'm going to show you how to do that. But before we talk more about STR and analysis, we have to talk about the technique to separate the DNA based on length. And that's called gel electrophoresis. You have to understand this. There is going to be a big question on the final test that requires you to understand how this works and be able to draw the results of an experiment. Okay? So pay attention to how this works. I'll try to explain it a little bit first, and then I'll show you a video, okay? Here, we have a chamber and the chamber is going to be filled with a solution. It's called buffer solution that helps conduct electric current. Okay, there's a slab of gel here. The gel is like, a little bit like the size of a phone here. Okay, like half of the phone, and it's like jello, right? You hold it up like a piece of little jello that's a little bit hardened, and you basically put the gel in a solution, and up here you're gonna have slots for you to load the DNA sample in. And when you run the current, when you hook up to a power supply and turn on the power, the current will go from negative to positive. And do you remember the charge of a DNA molecule? Anybody? Is DNA positive or negative? Think of the backbone. 50% chance. What's the backbone made up of? Sugar and phosphate. And what's the phosphate formula from chemistry class?
PO4.
Yeah, okay, almost, right? PO4 -3, right? Okay, so the backbone of the DNA is negatively charged. If you apply a current going from negative to positive, it will push the negative DNA down the gel. Okay? So it's a little bit abstract right now because you can't really see the whole setup, but I'll show you a video in a second and it will make more sense. All right, so key thing here, you're gonna load the DNA here. Okay, and then you're gonna apply the current, and the current's gonna push the DNA down the gel. That's the only thing you need to understand right now. Do you understand it? Okay. Let's take a closer look at the gel. The gel is like this if you look at it from the top. You see the slots? Okay, that's where you're gonna load the DNA. You literally put the DNA into the gel. All right, and then they're gonna move down. They're gonna move down the gel and follow the current. This gel is like it's exactly like making jello. Okay. Have you guys made jello before? Jello? Yeah. Comes in a package, right? Okay, you add water to it, right? That's exactly how you do it. It comes in a package, but like in a bottle more. It's powder. You put the powder in some special water solution, and then you microwave it, and then you let it, you pour it into like a cask, and then you let it cool. When it cools, it becomes a gel like that. Okay? I've done it, so I'm speaking from experience. You just have to believe me. Now, how do you make these little slots? After you pour the gel into the cask, you basically put a comb in it. Okay, so when the gel kind of solidifies around the comb, afterwards you pull the comb out, then you're gonna have slots in it. You'll see in the video. Now, if you zoom in into this gel, is gonna look like this under the microscope. It's basically a mishmash, a network. Okay, and so when you force the DNA to go through this network, the long DNA will get tangled up, and the shorter one will be able to weave through quite easily. Which means if the DNA is really long, they won't travel down the gel too far. But if it's really short, then they're going to be able to move through very quickly. All right. It's like this thing, like I saw at the playground with my kids, and I thought of this electrophoresis, so I took a picture of it. If you haven't saw this before, what happens is like you get on the top, and then you try to get to the bottom. All right. So like my kids was like challenged me, right? So I'll race you to the bottom. I said, sure, yeah, let's do it, right? But it was actually quite hard because I'm bigger than my kids, right? Obviously, and then I get tangled up. So by the time you get to the bottom, I'm only up here. And that's what happens with the DNA separation. If it's really big, if it's really long, it's not like it cannot get through. It just needs more time to do it, right? So if you only give it like 1 hour to run down the gel, the longer DNA will get stuck at the top, while the shorter ones, smaller ones, will be able to get through very quickly. Is that okay? All right. So this gel, all right, it's like racing, okay, race horse or something like that. Every sample has its own lane. Okay, so that's one sample, and then it's gonna run down its lane. It's not gonna go sideways or anything like that. Okay, it's just gonna keep on going down like that. Okay, it's like racing down the lane. Right? We good so far? All right. I'm gonna show you a video, but then I cannot record it because YouTube is gonna say like I copyright or whatever, and then it's not gonna let me upload the video. So I'm gonna have to pause this. So in order to see the DNA, you have to add a special compound called ethidium bromide. Ethidium bromide binds to DNA and it glows, it fluoresces under UV light. Okay, so you take the gel out of the chamber when it's done, and then you soak it in ethidium bromide, and you wait for the compound to bind to the DNA. Because this stuff binds to DNA, it's carcinogenic, okay? So you actually have to wear gloves and be careful not to get any on yourself. And then afterwards, you expose the gel to UV light, and then it will show you all the locations where DNA exists. Okay, now it's a little bit hard to tell, 3D like this, but usually you can take like a black and white picture like this, and you can see, okay, so there is a DNA sample, there is one, right? So remember, this is sample one, sample two, so on and so forth, right? And which one is longer? 3 here is longer or 4 is longer? This is the top of the gel, right? They're going down. Is 3 longer or 4 longer? Because sample in number 4 didn't travel as far down, right? Right? That means it must be longer. The shorter one is always going to be further down. But how do you quantify these things? Like, what if I actually want to know the exact measurement? Well, you can buy these pre-mixed DNA samples. They are called a ladder. It's a little bit hard to see with the lights on. You see there are light bands here? You see it very faintly? So that's called ladder. DNA ladder. And the ladder is pre-created DNA with known length, right? So there's usually like a little reference that comes with the ladder, and then they'll be like, oh, this line right here is about 260 or 270 here. So you will know this one is about 268. Okay, so basically the ladder provides a reference for you to check the length. Okay, so let's do an example here. There's a crime scene DNA here, and we separated the length of the sample, the STR of the sample, basically. And then we have different suspects. And remember, each band represents the length of a specific STR, right? Yours is going to be different from mine, right? So which suspect was at the crime scene? Yeah, it's not rocket science, right, guys? It's number 2, right? Okay, because it's the same. All right. So there's no denying he or she was there, guaranteed. Okay, but whether they committed the crime or not, this won't tell you. This will only tell you that they're there. Got it? Okay. Now let's think about this. You will see questions like these on the final test. The plasmid is treated with equal R1 and BAM-H1 when the digested plasmid is separated using gel electrophoresis. How many bands will you see? Okay, so this is the plasmid. If I treat it with equal R1, we're going to cut it right here. And then we're going to cut it, where else? Right here, right? Right? So if you have a circle and you cut it twice, how many bands are you going to see? You guys use a rubber band if you can't see this, right? How many? Show me. 2 or 3? Like, give me a second, okay? You're gonna cut here and you're gonna cut here, right? So you're gonna get one piece and then you're gonna get a second piece. Make sense? Okay? So you're gonna see 2 bands. 2 bands. The longer band here is going to be closer to the top of the gel. And then the shorter one here is going to be further down the gel. Okay. I need you to understand this. This is linear DNA. One cut equals to how many fragments? Two fragments. This is circular. One cut equals to how many fragments? That's right. Understand? All right, let's take a look at this. Show me your answer. Yes, Tara. Very good. Anyone else? Good. Good. Very good. Okay, look. Sample A is CAG repeated 10 times. How long is that? 30. CAG repeated 15 times. Yeah, I mean, like it's the same thing. You don't actually have to calculate the actual length. 15 is more than 10, right? Okay. So this is shorter, and then this is longer. Which one will be further down the gel? Shorter ones always go further, right? That's why the answer is A. Got it? Here we have a DNA letter, and then we have a sample that's being treated with restriction enzyme. Okay, so basically, it says it's a linear DNA, and then you cut it. How many times does it say? It only has two recognition sites. Okay, I guess it does. So that means like you have linear, you cut it twice, and then we have three pieces, right? Okay, and the three pieces are gonna be different length, right? And you're gonna run this on the gel. You're gonna have three fragments. So now, how long is the original? That's what the question is asking. So this is the ladder, right? The reference, the ruler, if you would. So this is 6 kilo bytes, as in kilobase rather. So like 6,000 base long. So that's 6, right? 6. How about this one? 9, right? It's between 10 and 8 is 9. And so 9 plus 6, 15. How about this one? 2.5, exactly, right? So 9 plus 6 plus 2.5. What is that? Yeah, 17.5 kb. That's all. Okay, I have to give you a note now. Oh, I only have 20 minutes. My God. Uh, let's try to finish this as best as I can. It would be best if you have 3 colors, guys. Okay, so this is Bob right here. And theoretically, let's say on chromosome uh 10, Bob has an STR right here. And it's gonna be like CCT repeated 20 times, okay? CCT repeated 20 times. And then over here, oh, that looks very similar. Let's over here on another chromosome, he has AT repeated 5 times. And this is chromosome, 17. And then on chromosome 20, he's gonna have another CGG maybe, and this is gonna be repeated 10 times. All right, over here we have another person, Alice. She's also human, which means on her chromosome 10, 17, and 20, she will have the same STRs. Chromosome 10, bless you. Now, they are not related, so she might have a CCT that's only repeated 20 times. Does everybody understand so far? Like what these lines are? You guys get it? Those are the polymorphism, right? The short tandem repeats. Everybody has it. The nucleotides don't change. What changes is the length, right? So like Bob's repeated 20 times over here. Oh, I see why. I used the same number, didn't I? Maybe that's why you guys are confused. So let's use something else. What should I use? 25 maybe? 25? Next time if I make a mistake, just tell me right away, okay? Don't wait until I discover it. Okay, and then this one maybe 80. It's the same. Okay? It happens to be the same. Maybe they both have the same ethnicity, right? So, they share more commonalities than people who are of a different race. And then here we have chromosome 20. And then let's just say this is CGG repeated 5 times. I think we are okay. Now, remember, because of the Human Genome Project, we know the exact location of all these STRs in the genome, which means if I take a sample of a DNA sample from Bob, and like just do a cheek swab or something like that, we can design primers that would specifically clone the STR. So I would find a primer that is before and after the STR, and then it will copy everything in between. I will have another set of primer that copies all this blue stuff and then all these green stuff. Right, so in the end, what you're gonna have is okay, we're gonna clone these STRs, short tandem repeats, with PCR. And then you're gonna have a sample in one of those tube things, and then it's gonna have, the red DNA, the blue DNA, and then the green DNA. Okay, same thing over here. Along these STRs, you have to be comparing the same set of STRs with PCR. And again, you're going to have a tube that has a mixture of them. Okay, and then I'm going to have a gel here. Okay, to draw the gel, it's like drawing a castle top. Now that's a slot for loading the sample. Negative at the top, positive at the bottom. This is the current. Oh, just gotta write it normally. So let's say I load Bob's sample here. Now, again, remember when you run it, you can't see the DNA, you have to stain it. And after you stain it, everything is just black and white, okay? But I'm going to use color to help you understand which one goes with. So let's do some math here together. This is CCT repeated 20 times. So how long is the entire length of this STR? 60, okay? So you can write it down if you want. This is 60. This is two times 5, which is 10, and then this is 30, right? So which one is going to be closest to the top? Red, blue, or green? Remember, you load the sample here. Which one is going to be closest to the top after you run the gel? When I say run the gel, I mean send the current through. Red, blue, or green? Red, exactly. Because it's the longest one, right? Okay. So it's going to be like somewhere up here. Furthest down, is it going to be blue or green? Blue is the shortest, so it's going to go furthest down. You're going to be able to weave through the fibers much quicker than the other ones. And green is going to be somewhere in between. Now, this is not like a linear relationship, okay? So it's not like if 60 is up here, 30 is going to be exactly halfway. Right? It's not like, it's not linear. Okay, so that's Bob right there. Now, I'm going to give you a moment to write, let's put Alice right here. Okay. Please draw Alice's 3 bands in relation to Bob's. Okay. Because look, if 60 is here and you have something like this one, how long is this one? 75, right? Okay. So is 75 going to be higher than Bob's red or below Bob's red? Higher, right? Exactly. All right, please do the other ones. Not bigger? Oh. All right, so the blue one, where will the blue one be? This blue one right here is going to be 10. Where is it going to go? Same as Bob's, right? Yeah, because Bob's also 10, right? So there we go. And then for the last one here is 3 times 5 is 15. So 15 is very close to 10. It's just a little bit longer, so it's going to be very close to it. Okay? So you see, with 3 locations of STR, it's already different. And you can tell them apart. But if we actually gonna do this for real, we're not gonna just use 3 locations. Because 3 is not enough to distinguish billions of people on the planet, right? There are like 8 billion people on the planet. So we have to use a lot of these STRs. And when you do it all at the same time, you're gonna end up with a picture like this. Right? Each of these bands, again, some are fainter than other ones, but each band represent a particular STR. Okay, and here it's like a smear. That means it's like a bunch of them really close, like maybe, like 10, 15, 20. It's really close that the gel is not going to be able to separate them. Okay, but that's how you create a DNA profile of a person. Do you guys understand? Yeah. So I'm not going to ask you to describe that, but you need to know how to draw the bands when I ask you to do it. Okay, we're not gonna have time to go through this RFLP. It's not gonna be on the test. We have a little bit of time. Maybe we try to do this one together. Something like this is like this particular one's not gonna be on the test, okay? But it shows you how we can apply some of these things. So we have a family of 4 here, 2 parents, 2 kids. Can we figure out who are the parents and who are the kids? That's the thing, right? So let's take a look here. Right. B, you know what? I gotta do it on over here. You guys don't have this, right? Yeah, that's because it's not on the test, okay? But I'll show you as an example, okay? So 2 parents, 2 kids. B is the child of A and C. Is that possible? So let's say this is the parents and this is the parents. So B is the child. That means everything B has, each of these DNA STR that B has to come from mom or dad, right? So let's take a look. This one is okay. It came from C. Okay. This one is okay. It came from C. This one, not okay. A doesn't have it. C doesn't have it. Where did B get it from? No one. That means A and C is not the parents of B. Do you understand? Okay, so this is no. Is C the child of A and B? Okay, you go figure it out. I'll make it bigger. Yes? Yeah, okay, let's see, right? Is this one okay? This one came from B, right? This one came from A, B, A, A, B, A, B, A, A or B, right? Okay, we don't know, right? Okay. So yeah, that's possible. Yeah. I mean, it's not definitive, but it's possible, right? And then you can check the other ones as well, right? So it turns out all the other ones are wrong. Right? So this is correct. You understand? Right? So that's how they actually do it in real life. Right? If they want to figure out, are these your kids or not, right? Because you heard about stories where people lost one of their kids, and then 10, 20 years later you find them, but then like you're not really sure if they're your kids. I saw this Netflix. Let me stop recording so it doesn't sound like I'm rambling. I saw this Netflix